Welcome to our channel. Today we are going to discuss cellular messengers in humans. Let's get start. Cellular messengers. These are molecules responsible for transmitting signals within or between cells in the human body. These messengers regulate physiological processes and maintain cellular homeostasis by relaying information from one part of the body to another. They can be classified based on their origin, function, or mechanism of action. Types of cellular messengers. Cellular messengers are broadly categorized into the following types. A. Intracellular messengers. These operate within the cell and relay signals from receptors to their target molecules in the cytoplasm or nucleus. Its examples are cyclic adenosine monophosphate, CAMP. Cyclic adenosine monophosphate, CAMP, is a crucial intracellular secondary messenger involved in transducing signals from extracellular stimuli, such as hormones and neurotransmitters, to regulate a wide range of physiological processes. Structure of CAMP. CAMP is a cyclic nucleotide derived from adenosine triphosphate, ATP. The structure consists of an adenine base, R, ribose sugar, and a phosphate group in a cyclic configuration. Synthesis of CAMP. The synthesis of CAMP is stimulated by trophic hormones that bind to receptors on cell surface. CAMP levels reach maximal levels within minutes and decrease gradually over an hour in cultured cells. Cyclic AMP is synthesized from ATP by adenylate cyclase located on the inner side of the plasma membrane and anchored at various locations in the interior of the cell. Adenylate cyclase is activated by a range of signaling molecules through the activation of adenylate cyclase, stimulatory Gs, protein-coupled receptors. Adenylate cyclase is inhibited by agonists of adenylate cyclase inhibitory GI, protein-coupled receptors. Liver adenylate cyclase responds more strongly to glucagon, and muscle adenylate cyclase responds more strongly to adrenaline. Degradation of CAMP. Enzyme phosphodiesterase, PDE, converts CAMP to AMP, terminating its signaling activity. PDE inhibitors, for example caffeine, theophylline, prolong CAMP action. Mechanism of action. 1. Activation of protein kinase A, pKa. CAMP binds to the regulatory subunits of pKa, causing the release of its catalytic subunits. Active pKa phosphorylates specific target proteins, altering their function. Hairy we can see after binding to its cell membrane beta-adrenergic receptor, the ligand, epinephrine, produces different effects in target cells by first stimulating production of CAMP, which converts inactive protein kinase A, pKa, to active pKa-. In liver and skeletal muscle, pKa phosphorylates the enzyme glycogen synthetase, converting it from an active to an inactive form and thus reducing glycogen synthesis, which is not shown here. pKa* converts inactive enzyme phosphorylase B through an additional phosphorylation to its active form, phosphorylase A*, and causes hydrolysis of glycogen to release glucose 1-phosphate, G1P, which in liver can be converted to free glucose and free phosphate, P. Free glucose leaves the cell via mediated transport. AC, adenylyl cyclase. CAMP can also affect directly by regulating ion channels, for example cyclic nucleotide-gated channels, and exchange proteins, for example EPAC. Physiological roles of CAMP. Metabolic regulation. Glucose homeostasis stimulates glycogen breakdown in the liver by activating glycogen phosphorylase and inhibiting glycogen synthase. Promotes lipolysis in adipose tissue by activating hormone-sensitive lipase. Cardiovascular system. Enhances heart rate and contractility by modulating calcium channels in cardiac myocytes. Mediates relaxation of smooth muscles in blood vessels, leading to vasodilation. Nervous system. Involved in memory and learning by modulating synaptic plasticity. Regulates neurotransmitter release. Endocrine functions. Acts as a mediator for hormones like glucagon, adrenaline, and parathyroid hormone, PTH. Stimulates steroidogenesis in adrenal and gonadal tissues. Immune response. Modulates inflammation by influencing cytokine production and immune cell activity. Renal function. Regulates water reabsorption in the kidneys by mediating the effects of antidiuretic hormone, ADH. Pathways. Involving CAMP. 1. Beta-adrenergic receptor signaling. Adrenaline binds to beta-adrenergic receptors, activating adenylate cyclase via G's proteins and increasing CAMP levels. 2. Glucagon signaling. Glucagon binding to its receptor elevates CAMP, promoting glycogenolysis and gluconeogenesis. 3. ADH signaling. ADH increases CAMP levels in kidney collecting ducts, enhancing water reabsorption via aquaporin channels. Clinical relevance. Diseases associated with CAMP. Dysregulation. Heart failure. Impaired CAMP signaling affects cardiac contractility. Diabetes. Mellitus. Altered CAMP pathways impact glucose metabolism. Asthma. Dysregulated CAMP in airway. Smooth muscles contributes to bronchoconstriction. Therapeutic applications. Beta agonists. Increase CAMP levels in smooth muscle cells to relieve bronchospasm in asthma. PDE inhibitors. Used in conditions like heart failure, Milrinone, and erectile dysfunction, sildenafil. Cyclic guanosine monophosphate, CGMP, cyclic guanosine monophosphate, CGMP, is a critical intracellular secondary messenger that regulates various physiological processes, such as smooth muscle relaxation, visual phototransduction, and cardiovascular homeostasis. It is synthesized in response to specific extracellular signals and works primarily through protein kinases, ion channels, and phosphodiesterase. Structure of CGMP. Chemical composition. CGMP is a cyclic nucleotide derived from guanosine triphosphate, GTP. Structure. It consists of a guanine base, a ribose sugar, and a phosphate group in a cyclic configuration. Degradation of CGMP. Enzyme. Phosphodiesterase. PDE. Converts CGMP to GMP, terminating its signaling activity. PDE5, a specific isoform, is involved in degrading CGMP in vascular smooth muscle and is a therapeutic target for certain drugs. Physiological roles of CGMP. Smooth muscle relaxation. CGMP mediates vasodilation by reducing intracellular calcium levels in vascular smooth muscle cells. Important in regulating blood pressure and blood flow. Cardiovascular system. CGMP is involved in heart rate modulation and cardiac output. Regulates the effects of natriuretic peptides, promoting diuresis and natriuresis. Visual phototransduction. cGMP plays a key role in rod and cone photoreceptor cells in the retina. Controls the opening and closing of cyclic nucleotide-gated ion channels, crucial for light signal transduction. Renal function. cGMP mediates the effects of ANP and BNP, leading to increased sodium excretion and reduced blood volume. Platelet aggregation. Inhibits platelet aggregation by reducing calcium levels, thus preventing thrombus formation. Gene transcription. cGMP binds to PKG, activating it. PKG phosphorylates specific target proteins, some of which are involved in regulating transcription. PKG can influence the activity of transcription factors such as CREB, CAMP response element binding protein, by phosphorylating co-activators or upstream kinases. This leads to altered gene expression related to cell growth, survival, or metabolism. Mechanism of action. 1. Activation of protein kinase G, PKG, cGMP binds to package, causing a conformational change and activation. PKG phosphorylates target proteins, altering cellular functions. 2. Regulation of ion channels. cGMP directly regulates ion channels, such as cyclic nucleotide-gated channels in the retina. 3. Interaction with PDEs. cGMP modulates its own degradation by activating certain PDEs. Calcium ions, Ca², as cellular messengers in humans. Calcium ions, Ca², are universal and versatile intracellular messengers involved in regulating various cellular processes, such as muscle contraction, neurotransmitter release, enzyme activity, and gene expression. The ability of Ca² to act as a signaling molecule arises from its highly regulated cytosolic concentration and interaction with specific binding proteins. Key features of calcium as a messenger. Resting cytosolic calcium levels. The intracellular Ca² concentration is maintained at very low levels, approximately 100 nm, compared to extracellular levels, approximately 1 to 2 mm. This steep concentration gradient allows for rapid and significant changes in intracellular Ca² levels in response to stimuli. Calcium signals. Amplitude. The magnitude of Ca². Rise. Frequency. Oscillations or spikes in Ca² levels. Calcium waves. Duration. How long the calcium signal persists. These characteristics encode specific cellular responses. Sources of intracellular calcium. Extracellular sources are voltage-gated calcium channels, VGCCs, open in response to membrane depolarization, allowing Ca² influx. Ligand-gated calcium channels. Channels like N-methyl-D aspartate, NMDA. Receptors permit Ca² entry upon ligand binding. Intracellular calcium stores are endoplasmic reticulum, ER. Calcium is stored in the ER and released via inositol trisphosphate, iodine triphosphide, receptors or ryanodine receptors in response to signals. Mitochondria. Mitochondria sequester calcium for buffering and signaling. Calciosomes. Specialized organelles that act as calcium storage depots to maintain cellular homeostasis and prevent toxic calcium levels. Intracellular calcium is actively removed or sequestered after signaling. Plasma membrane calcium ATPase, PMCA, pumps calcium out of the cell using ATP. Sodium calcium exchanger, NCX, exchanges intracellular calcium for extracellular sodium. SERCA pumps, sarco, endoplasmic reticulum calcium ATPase, pumps calcium back into the ER. Mitochondrial uptake. Mitochondria sequester excess calcium for buffering. Mechanisms of calcium signaling. First step is calcium entry from extracellular space. Calcium influx is triggered by external stimuli, such as membrane depolarization or ligand binding. Mechanisms include voltage-gated calcium channels, VGCCs, open during membrane depolarization, primarily in excitable cells, for example neurons, muscle cells. Ligand-gated ion channels, activated by neurotransmitters or other ligands. Examples are N-methyl D-aspartate, NMDA, receptors, nicotinic, acetylcholine receptors. Store-operated calcium entry, triggered by depletion of calcium stores in the ER. Mediated by STIM-1, calcium sensor in the ER, and/or I1, calcium channel in the plasma membrane. Second step is intracellular calcium release. Calcium stored in organelles, such as the endoplasmic reticulum, ER, and mitochondria, is released in response to intracellular signals. Its mechanisms are inositol trisphosphate, iodine triphosphide pathway. Hormones or neurotransmitters activate G-protein coupled receptors, GPCRs, stimulating phospholipase C. PLC. PLC cleaves PHOSPHATIDY LINOSITOL4, 5-bisphosphate, PIP, to generate IP. Iodine triphosphide binds to iodine triphosphide receptors on the ER, causing calcium release. Calcium-induced calcium release. CICR. Small increases in cytosolic calcium ions trigger larger releases from ryanodine receptors. RYRs, in the ER or sarcoplasmic reticulum, SR. NAADP. Nicotinic acid adenine dinucleotide. Phosphate. Activates calcium release from lysosomes or endolysosomal compartments via two-pore channels, TPCs. Excess calcium is removed by plasma membrane calcium ATPase. PMCA. Sodium calcium exchanger, NCX, and SERCA pumps. Calcium binding proteins. Calmodulin, a key calcium binding protein that undergoes conformational changes upon binding Ca². Activates various target enzymes, such as Ca², calmodulin-dependent protein kinases, CAMKs. Troponin C. Regulates muscle contraction by interacting with actin and myosin in response to Ca². Parvalbumin and calbindine buffer intracellular calcium to regulate its levels. Functions of calcium as a messenger. 1. Muscle contraction. Ca² binds to troponin, causing conformational changes that enable actin-myosin interaction and contraction. 2. Neurotransmitter release. In presynaptic neurons, Ca² influx triggers vesicle fusion and neurotransmitter release. 3. Gene expression. Ca² activates transcription factors like NFAT, nuclear factor of activated T cells, and CREB, camp response element binding protein, to regulate gene expression. 4. Cellular metabolism. Regulates enzymes like pyruvate dehydrogenase in mitochondria, affecting ATP production. 5. Apoptosis. Prolonged elevation of Ca² can activate pathways leading to programmed cell death. 6. Cell cycle and proliferation. Calcium signaling. Modulates the activity of cyclins and cyclin-dependent kinases, CDKs. 7. Immune response, Ca². Signaling is essential for T cell activation and function. 8. Fertilization, calcium waves in the oocyte following sperm entry are critical for activation of embryonic development. Regulation of calcium signaling. Temporal and spatial control. Calcium signals are tightly regulated to ensure specificity of response. Calcium oscillations. Oscillatory patterns convey distinct signals to the cell, influencing processes like gene expression. Feedback mechanisms. Calcium-sensitive enzymes and proteins modulate its own signaling pathways. Pathological implications. Hypocalcemia, low extracellular calcium levels disrupt signaling, leading to muscle spasms, tetany, and cardiac arrhythmias. Hypercalcemia. Excessive calcium causes toxicity, kidney stones, and calcification of soft tissues. Neurological disorders. Dysregulated calcium signaling is implicated in Alzheimer's disease, epilepsy, and neurodegeneration. Cardiac diseases. Abnormal calcium handling contributes to arrhythmias and heart failure. Inositol triphosphate, iodine triphosphide, and diacylglycerol, DAG. These are two critical second messengers in human cells that mediate intracellular signaling. These molecules are generated during signal transduction processes initiated by the activation of certain cell surface receptors, such as G-protein-coupled receptors, GPCRs, and receptor tyrosine kinases, RTKs. Steps in the synthesis of iodine triphosphide and DAG. First step in signal initiation. The synthesis of iodine triphosphide and DAG begins with the activation of specific cell surface receptors by extracellular ligands. Key receptors. G-protein-coupled receptors, GPCRs. Activated by hormones, neurotransmitters, or other signaling molecules. Activate the GQ protein, which in turn activates PLC. Receptor. Tyrosine kinases, RTKs. Activated by growth factors like epidermal growth factor, EGF, or platelet-derived growth factor, PDGF. Directly activate PLC through phosphorylation. Second step is activation of phospholipase C, PLC. Once activated, PLC hydrolyzes phosphatidylinositol 4,5-bisphosphate, PIP, a phospholipid present in the inner leaflet of the plasma membrane. Last step is cleavage of PIP, phosphatidylinositol 4,5-bisphosphate, PIP, is cleaved by activated PLC into inositol 1,4,5-trisphosphate, iodine triphosphide, a water-soluble molecule that diffuses freely in the cytoplasm. Diacylglycerol, DAG, a lipid-soluble molecule that remains anchored in the plasma membrane. Mechanisms of action of inositol triphosphate and diacylglycerol. Mechanism of action of inositol triphosphate. Iodine triphosphide binds to iodine triphosphide receptors, IPRS, which are calcium channels located on the membranes of the endoplasmic reticulum. IPRS are ligand-gated ion channels activated specifically by IP. Calcium release. Iodine triphosphide binding induces a conformational change in IPRs, opening calcium channels and allowing the release of calcium ions, Ca², from the ER into the cytoplasm. Calcium signaling. The increase in cytoplasmic Ca² acts as a signal to activate various calcium-dependent processes, including enzymatic activation. Calcium-dependent enzymes like calmodulin-dependent protein kinases, CAMKs, and phosphatases. Cytoskeletal dynamics. Calcium influences actin and microtubule organization. Exocytosis triggers vesicle fusion with the plasma membrane in neurons and endocrine cells. High cytosolic Ca² levels inhibit IPRs to prevent excessive calcium release. Excess calcium is resequestered into the ER by the sarco-endoplasmic reticulum calcium ATPase, SERCA, or expelled by plasma membrane pumps. Termination. Iodine triphosphide is rapidly dephosphorylated to inositol or lower phosphorylated intermediates by inositol polyphosphate phosphatases. Calcium levels return to baseline as pumps restore homeostasis. DAG binds to the regulatory domain of protein kinase C, PKC, isoforms, recruiting them to the plasma membrane. PKC activation requires DAG and, in some cases, calcium ions, Ca², released via iodine triphosphide signaling. Once activated, PKC phosphorylates target proteins involved in cell proliferation, modulation of mitogenic signaling pathways, gene expression, regulation of transcription factors like NF-kappa B, apoptosis, influence on cell survival mechanisms. DAG also activates other proteins, such as Ras-guanyl nucleotide releasing proteins, RAS-GRPs, which regulate small GTPase signaling. Termination. DAG is metabolized by diacylglycerol kinase, DAGK, into phosphatidic acid, PA, or hydrolyzed by lipases. IP-mediated calcium release and DAG-mediated PKC activation often occur in parallel and/or interdependent. Calcium ions enhance the activation of certain PKC isoforms, amplifying the signaling response. Physiological roles of iodine triphosphide and DAG. 1. Regulation of calcium homeostasis. IP-mediated calcium release is essential for processes such as muscle contraction, exocytosis, and fertilization. 2. Activation of protein kinase C, PKC. DAG-dependent PKC activation plays a role in cell proliferation, differentiation, and survival. 3. Immune responses. Both iodine triphosphide and DAG signaling contribute to T-cell receptor TCR signaling, enabling T-cell activation. 4. Neurotransmission. IP-induced calcium release modulates synaptic activity and neurotransmitter release. 5. Hormonal regulation. DAG and IP mediate responses to hormones such as vasopressin, oxytocin, and angiotensin II. Pathological implications of iodine triphosphide and DAG dysregulation. 1. Cancer. Overactivation of DAG-PKC signaling is implicated in tumor growth and survival. 2. Neurological disorders. Altered iodine triphosphide signaling contributes to neurodegenerative diseases like Alzheimer's. 3. Cardiovascular diseases. Dysregulated DAG-PKC activity can affect vascular tone and lead to hypertension. 4. Immune dysregulation. Abnormal IP-DAG signaling is associated with autoimmune diseases and inflammation. Nitric oxide as a cellular messenger in humans. Nitric oxide, NO, is a unique and versatile signaling molecule involved in a variety of physiological and pathological processes in humans. As a gaseous and freely diffusible messenger, NO operates differently from traditional second messengers like calcium or cyclic nucleotides. It plays critical roles in vascular regulation, immune responses, neurotransmission, and more. Synthesis of nitric oxide. Nitric oxide synthase, NOS, enzyme mediates the conversion of L-arginine to L-citrulline and nitric oxide. Using reduced nicotinamide adenine dinucleotide phosphate, NADPH, as a cofactor. Here we can take endothelial cells. Nitric oxide is synthesized by endothelial nitric oxide synthase, which diffuses through the membrane and activates SGC, which in turn increases cGMP that leads to vasorelaxation. LNMMA, ADMA and SDMA compete with L-arginine and reduce NO synthesis. Isoforms of NOS. 1. Endothelial NOS, expressed in endothelial cells. Regulates vascular tone and blood flow. 2. Neuronal NNOS, found in neurons. Functions in neurotransmission and synaptic plasticity. 3. Inducible NOS, induced in immune cells like macrophages during inflammation. Produces large amounts of NO for defense mechanisms. Mechanisms of nitric oxide. Action. Nitric oxide acts as a paracrine and autocrine messenger, diffusing freely across membranes. Its effects are mediated by two primary mechanisms. 1. Activation of guanylyl cyclase. NO binds to the heme group of soluble guanylyl cyclase, SGC, in target cells. This activates SGC, catalyzing the conversion of guanosine triphosphate, GTP, to cyclic guanosine monophosphate, cGMP. cGMP functions as a second messenger to regulate downstream pathways, including vasodilation. cGMP activates protein kinase G, PKG, leading to smooth muscle relaxation and reduced vascular resistance. Platelet inhibition prevents aggregation and clot formation. Neurotransmission modulates neuronal signaling via cGMP-dependent pathways. And gene expression. 2. S-nitrosylation of proteins. NO reacts with thiol groups in proteins, forming S-nitrosothials. This post-translational modification alters protein function and regulates enzyme activity. Ion channel conductance. Protein-protein interactions. Physiological roles of nitric oxide. 1. Vascular system. Vasodilation. NO is produced by endothelial cells in response to shear stress or signaling molecules like acetylcholine. It diffuses to smooth muscle cells, where it activates SGC, leading to muscle relaxation and blood vessel dilation. Blood pressure regulation. NO helps maintain normal blood pressure by reducing vascular resistance. 2. Immune system. Host defense. NO produced by iNOS in macrophages acts as a cytotoxic agent against pathogens. Regulation of inflammation modulates the activity of immune cells and inflammatory mediators. 3. Nervous system. Neurotransmission. NO acts as a retrograde messenger in the brain, modulating synaptic plasticity, learning, and memory. Smooth muscle relaxation. NO released from nitrogen neurons regulates gastrointestinal and urogenital smooth muscle function. 4. Respiratory system. Regulates airway tone and facilitates oxygen delivery to tissues. 5. Reproductive system. Involved in penile erection by promoting vasodilation in the corpus cavernosum. 6. Cellular metabolism. Modulates mitochondrial respiration and energy production. Regulation of nitric oxide signaling. Enzyme expression, NOS and NNOS, are constitutively expressed but activated by calcium, calmodulin. NOS is induced during inflammatory responses. Cofactor availability. Adequate levels of cofactors like BHR. Critical for NOS activity. Degradation. NO is short-lived and rapidly reacts with oxygen or superoxide to form reactive nitrogen species, for example, peroxynitrite, limiting its activity. Feedback mechanisms. High levels of NO inhibit NOS activity through negative feedback loops. Pathophysiological implications of nitric oxide. Cardiovascular diseases. Endothelial dysfunction. Reduced NO production or bioavailability contributes to hypertension, atherosclerosis, and coronary artery disease. Shock. Excessive NO production during septic shock leads to severe hypertension. 2. Neurodegenerative disorders. Dysregulated NO signaling is implicated in Alzheimer's disease, Parkinson's disease, and stroke. 3. Cancer. NO has dual roles in cancer. Low levels promote angiogenesis and tumor growth. High levels induce apoptosis and inhibit tumor progression. Inflammatory diseases. Overproduction of NO by NOS contributes to chronic inflammation in conditions like rheumatoid arthritis and inflammatory bowel disease. Pulmonary disorders. Impaired NO signaling leads to conditions like pulmonary hypertension and asthma. Hormones are cellular messengers in humans. Hormones are biochemical substances secreted by specialized cells, primarily endocrine glands, to regulate physiological processes in the body. They act as cellular messengers by binding to specific receptors on target cells, initiating signal transduction pathways, and eliciting specific biological responses. Hormones are critical for maintaining homeostasis, regulating growth, development, reproduction, metabolism, and stress responses. Classification of hormones. Hormones are classified based on their chemical structure and solubility. 1. Peptide and protein hormones. Composed of chains of amino acids. Examples are insulin, glucagon, growth hormone. These hormones bind to cell surface receptors as they cannot cross the lipid bilayer. 2. Steroid hormones. Derived from cholesterol. Examples are cortisol, aldosterone, testosterone, estrogen. These hormones diffuse across the cell membrane and bind to intracellular receptors. 3. Amino acid-derived hormones. Synthesized from tyrosine or tryptophan. Examples are thyroid hormones, TNT, epinephrine, norepinephrine, melatonin. These hormones act through both surface and intracellular receptors, depending on the hormone. 4. Lipid-derived hormones, eicosanoids, derived from fatty acids, for example arachidonic acid. Examples are prostaglandins, thromboxanes. These hormones bind to specific cell surface receptors. Hormonal signaling pathways. Hormones function as cellular messengers by initiating and propagating signaling pathways. The steps involved are: first is hormone secretion. Hormones are synthesized in endocrine glands and secreted into the bloodstream. Second step is transportation. Water-soluble hormones, for example peptide hormones, dissolve in blood plasma, while lipid-soluble hormones, for example steroid hormones, bind to carrier proteins for transport. Third step is receptor binding. Hormones bind to specific receptors. located either on the cell surface, for example G protein-coupled receptors, or inside the cell, for example, nuclear receptors. Fourth step is signal transduction. The receptor-hormone interaction triggers intracellular signaling cascades, such as activation of second messengers, for example C, AMP, IP, calcium ions, phosphorylation of target proteins, modulation of gene transcription. The cellular response occur. Target cells exhibit a response, such as enzyme activation or inhibition, alteration in gene expression, changes in ion channel activity. Last step is termination. Hormonal signaling is terminated by hormone degradation, receptor desensitization, or signaling pathway inhibition. Modes of hormonal communication. Hormones communicate using the following mechanisms. 1. Autocrine signaling. Hormones act on the same cell that produce them. Example, tumor cells producing growth factors for self-proliferation. 2. Juxtacrine signaling. Hormones act on adjacent cells through direct contact. Example, Notch signaling in cell differentiation. 3. Paracrine signaling. Hormones act on neighboring cells in the same tissue. Example, prostaglandins in inflammatory responses. 4. Endocrine signaling. Hormones are secreted into the bloodstream and act on distant target cells. Example, insulin acts on muscle and liver cells to regulate glucose uptake. Examples of major hormonal pathways. 1. Insulin, peptide hormone, pathway. 2. Cortisol, steroid hormone, pathway. 3. Thyroid hormones, T and T. 4. Epinephrine, amino acid-derived hormone pathway. 5. Prostaglandins, lipid-derived hormone, pathway. Dysregulation of hormonal signaling. Abnormal hormone signaling can result in various diseases. Endocrine disorders, hyperthyroidism, imbalance in thyroid hormones, diabetes mellitus, insulin deficiency or resistance. Cancer, hormone-dependent cancers, for example, breast cancer linked to estrogen signaling. Reproductive disorders, polycystic ovary syndrome, PCOS, due to hormonal imbalance. Growth disorders, dwarfism, or gigantism due to growth hormone abnormalities. Neurotransmitters. Neurotransmitters are chemical messengers used by neurons to communicate with each other or with other types of target cells, for example muscle cells or glands. They play a pivotal role in transmitting signals across synapses, enabling functions such as thought, emotion, movement, and homeostasis in the human body. Types of neurotransmitters. Neurotransmitters are categorized based on their structure and function. 1. Amino acid neurotransmitters. Examples: glutamate, gamma-aminobutyric acid, GABA, glycine. Function: glutamate is excitatory, while GABA and glycine are inhibitory. 2. Peptide neurotransmitters. Examples: substance P, endorphins, enkephalins. Function: modulate pain perception and stress response. 3. Monoamines. Examples: dopamine, serotonin, norepinephrine, epinephrine. Function: involved in mood regulation, arousal, and autonomic functions. 4. Acetylcholine. Unique neurotransmitter involved in both central and peripheral nervous systems. Function: critical for muscle contraction, attention, and memory. Synthesis and release. Synthesis. Neurotransmitters are synthesized in the neuron's soma, or axon terminals. Enzymes specific to each neurotransmitter aid in synthesis. For example, dopamine is synthesized from tyrosine via tyrosine hydroxylase and dopa decarboxylase. Neurotransmitters are stored in synaptic vesicles within the presynaptic neuron. The release of neurotransmitters is triggered by an action potential arriving at the presynaptic terminal. Steps of neurotransmitter release. First step is the action potential arrival. Depolarization of the presynaptic membrane opens voltage-gated calcium channels. Second step is calcium influx. Calcium ions enter the neuron, increasing intracellular calcium concentration. Third step is vesicle docking and fusion. Synaptic vesicles are docked at the active zone of the presynaptic membrane. Proteins like snares, for example synaptobrevin, syntaxin. SNAP25, mediate vesicle fusion with the membrane. Fourth step is exocytosis. Vesicles release their contents into the synaptic cleft through exocytosis. Last step is diffusion. Neurotransmitters diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. Mechanism of action of neurotransmitters. The mechanism of action begins when neurotransmitters are released from presynaptic neurons and bind to receptors on postsynaptic cells. Receptors are specialized proteins that determine the cellular response and are classified into two main types. 1. Ionotropic receptors, ligand-gated ion channels. Function as ion channels that open or close in response to neurotransmitter binding. For example, glutamate receptors allow sodium influx, causing depolarization. GABA are receptors allow chloride influx, causing hyperpolarization. Its action is fast, milliseconds, and leads to immediate changes in the membrane potential. 2. Metabotropic receptors, G-protein coupled receptors, GPCRs, neurotransmitter binding activates intracellular signaling cascades via G-proteins. For example, dopamine receptors activate CAMP production. Muscarinic acetylcholine receptors influence ion channels indirectly. Action is slow, seconds to minutes, but modulates long-term cellular processes. Signal transmission. The cellular response depends on whether the neurotransmitter elicits excitation, inhibition, or modulation. Excitatory neurotransmitters cause depolarization of the postsynaptic membrane, lead to the generation of an excitatory postsynaptic potential, EPSP. For example, glutamate opens sodium-calcium channels, moving the membrane potential toward the threshold for action potential generation. Inhibitory neurotransmitters cause hyperpolarization of the postsynaptic membrane, lead to the generation of an inhibitory postsynaptic potential, IPSP. For example, GABA opens chloride channels, moving the membrane potential away from the threshold. Neuromodulatory neurotransmitters modulate synaptic activity without directly causing EPSPs or IPSPs. Influence neuronal excitability, synaptic plasticity, and receptor sensitivity. For example, dopamine regulates reward pathways and motor control. Termination of action. To maintain precise signaling, neurotransmitter action must be terminated quickly. This involves reuptake, in which transporters in the presynaptic membrane take up neurotransmitters. For example, serotonin reuptake via SERT, enzymatic degradation, in which enzymes break down neurotransmitters in the synaptic cleft. For example, acetylcholine degradation by acetylcholinesterase. Some neurotransmitters diffuse away from the synaptic cleft and are metabolized elsewhere. Cytokines. Cytokines are small proteins or glycoproteins that function as crucial signaling molecules in the immune system and other biological processes. They mediate communication between cells to regulate immunity, inflammation, hematopoiesis, and tissue repair. Cytokines act through specific receptors on target cells, triggering intracellular signaling pathways that modulate gene expression and cellular responses. Properties of cytokines. 1. Pleiotropy. A single cytokine can affect multiple cell types and elicit diverse responses. 2. Redundancy. Different cytokines can produce similar effects. 3. Synergy. Cytokines can work together to enhance responses. 4. Antagonism. Some cytokines can inhibit the action of others. 5. Autocrine, paracrine, and endocrine. Action. Autocrine act on the same cell that secretes them. Paracrine act on nearby cells. Endocrine act on distant cells via the bloodstream. Classes of cytokines. Cytokines are classified based on their function, structure, or receptor families. 1. Interleukins. ILs. Interleukins, ILs, are a group of cytokines primarily produced by white blood cells, leukocytes, and other immune cells. They play critical roles in regulating immune responses, inflammation, hematopoiesis, and tissue repair. Interleukins act as signaling molecules that mediate communication between immune cells and influence cell behavior through specific receptors. For examples, IL-1 promotes inflammation and fever. IL-2 stimulates T-cell proliferation. 2. Tumor necrosis factor, TNF. TNF is synthesized as a membrane-bound precursor protein and cleaved by the enzyme TNF-alpha converting enzyme, TACE, to release its active soluble form. Stimuli for TNF production include bacterial lipopolysaccharides, LPS, viral infections, and other pro-inflammatory signals. Regulate apoptosis, inflammation, and immune responses. 4. Example, TNF-alpha, key mediator of acute inflammation. 3. Colony-stimulating factors. CSFs. Colony-stimulating factors, CSFs, are a group of glycoproteins that regulate the production, differentiation, and function of hematopoietic, blood-forming, cells in the bone marrow. These cytokines stimulate the proliferation and maturation of specific lineages of blood cells and are crucial for maintaining immune and hematopoietic homeostasis. For examples, GCSF promotes neutrophil production. GM-CSF stimulates granulocyte and macrophage production. 4. Interferons, IFNs, provide antiviral defense and modulate immune responses. 4. Examples, IFN alpha, beta, induce antiviral states in infected and neighboring cells. IFN gamma activates macrophages and enhances antigen presentation. Chemokines. Chemokines are a family of small cytokines or signaling proteins that play a critical role in immune system regulation by directing the migration of immune cells to sites of infection, inflammation, or injury. They are essential for immune surveillance, inflammatory responses, and tissue homeostasis. Examples, CCL2, MCP1. attracts monocytes. CXCL8, EL8, attracts neutrophils. Transforming growth factors. TGFs. Transforming growth factors, TGFs, are multifunctional cytokines that play critical roles in regulating cell proliferation, differentiation, growth, apoptosis, and extracellular matrix (ECM) production. TGFs are essential for embryonic development, immune regulation, wound healing, and maintaining tissue homeostasis. Dysregulation of TGF signaling is associated with cancer, fibrosis, and other diseases. Example: TGF-beta inhibits inflammation and promotes wound healing. Mechanisms of cellular messaging. A. Signal reception. Signals are initiated when a messenger binds to its specific receptor, which may be on the cell surface or inside the cell. Receptors include G-protein coupled receptors, GPCRs, involved in CAMP and IP3 pathways. Enzyme-linked receptors, such as receptor tyrosine kinases. Ion channel receptors regulate ion flow in response to neurotransmitters. Intracellular receptors, located in the cytoplasm or nucleus, binding to lipid-soluble messengers like steroid hormones. B. Signal transduction. Once the receptor is activated, it initiates a cascade of intracellular events. Amplification. A single signal molecule can result in a large-scale response due to the activation of multiple downstream molecules. Pathways include CAMP pathway. Activation of adenylyl cyclase converts ATP to CAMP, which activates protein kinase A, pKa. MAPK, ERK pathway. Transmits growth and stress signals to the nucleus. Calcium signaling, Ca² release triggers responses like muscle contraction or exocytosis. C. Signal termination. Cellular responses must be tightly regulated to prevent overactivation. Signal termination involves degradation of messengers, e.g., CAMP biphosphodiesterase, receptor desensitization or internalization, removal of messengers from the extracellular space. Examples of cellular messaging systems. Endocrine signaling. Hormones are released into the bloodstream and act on distant targets. Example: insulin regulates glucose uptake in muscle and liver cells. Paracrine signaling. Messenger molecules act on nearby cells. Example: growth factors like VEGF stimulating angiogenesis. Autocrine signaling. Cells respond to signals they produce themselves. Example: cancer cells releasing growth signals to sustain their own proliferation. Synaptic signaling. Neurotransmitters are released into the synaptic cleft to target nearby neurons or muscles. Example: acetylcholine in neuromuscular junctions. Clinical relevance. Diseases. Dysregulation of cellular messengers leads to conditions like diabetes, insulin signaling, cancer, growth factor signaling, and autoimmune diseases. Cytokine imbalance. Therapeutics. Drugs often target cellular messengers or their pathways. Examples: beta blockers block adrenaline signaling. NSAIDs inhibit prostaglandin synthesis. Thanks for watching.